All right. Hello, everyone. Welcome to our presentation on the hydroxyl group and BH4 interactions in the mechanism of the human enzyme phenylalanine hydroxylase, or for short, PAH. I'm Audrey Camarillo.
And I'm Kyla Tonko.
All right. So first, let's talk about why this presentation is important. So 1 in 33 babies every year are born with a birth defect. And while many birth defects can be caused by environmental factors such as smoking during pregnancy or certain foods that you eat, certain congenital disorders can also be inherited through genetics, which is much like the one we're going to talk about today. And many of these disorders are due to deficiencies of certain proteins or enzymes, which are essential to the body. And congenital disorders can range from being barely noticeable to being completely fatal to a child at birth. And some prevalent examples of these can include Down syndrome or cystic fibrosis, lots of examples that we know about. But the one we're going to talk about today is PKU. 1 in 10,000 newborns are born with phenylketonuria, which is PKU for short. And that is caused by a deficiency of the enzyme we're going to talk about, phenylalanine hydroxylase. This enzyme converts phenylalanine to tyrosine. And the issue with this disease is that there are no existing supplements for PAH. So people who have this disease have to limit their intake of phenylalanine, which can be pretty tough in these days because pretty much everything is artificially sweetened, and phenylalanine is found in artificial sweeteners. And as you can see here, artificially sweetened drinks actually include a warning label for phenylalanine. And the only existing cure for this is to maintain a diet free of phenylalanine, which, as I said, is very tough.
This leads us to talk about the importance of proteins. Our biomolecule, PAH, is an enzyme which is a specific type of protein that carries out biochemical reactions. Proteins perform almost every function in the cell, and just for a little bit of review, they have 4 structures: primary, which is just the amino acid sequence; secondary, the specific folds, whether it be alpha helices or beta sheets; the tertiary structure, which includes the 3D structure, such as the disulfide bonds or hydrogen bonding within the protein; and lastly, the quaternary structure. Not every protein does include a quaternary structure, however, ours does, which will be talked about a little bit later. But continuing on, are the basic building blocks of proteins, which are amino acids. They all have generally the same structure, but the one thing that makes it unique is each of its different R groups, which can be categorized into nonpolar, polar, polar charged, and other groups as well. And the 2 amino acids that we'll be focusing on in our presentation today are phenylalanine, which is the substrate to the enzyme, and tyrosine, which is the product of the reaction. It initially doesn't seem as if it would be as complicated as a mechanism because it's just an added hydroxyl group onto the tyrosine. However, the process is highly regulated, which is why PKU is such a complex disease. And it's extremely important because accumulation of phenylalanine can become toxic to the body. And additionally, tyrosine is a precursor for biosynthesis of certain neurotransmitters in the brain. And without those neurotransmitters, it can lead to a cognitive disability.
So here's our thesis. The enzyme phenylalanine hydroxylase with this cofactor BH4 catalyzes the reaction to convert L-phenylalanine to tyrosine. The enzyme contains binding sites for L-phenylalanine and BH4 through the amino acids tryptophan and histidine. These positively charged residues create a stable environment for L-phenylalanine to be converted into tyrosine. The deficiency of PAH causes an autoimmune disorder called phenylketonuria, which can cause mental disorders and seizures because of the lack of tyrosine in the brain.
So for the general overview of the function of the enzyme, PAH catalyzes the hydroxylation of L-phenylalanine into L-tyrosine. And as we go through the presentation, we'll continue to unveil deeper and more complex mechanisms of the reaction once all the interacting biomolecules are introduced. However, this is just the general overview of the reaction.
So the PAH structure is generally a tetramer. It contains two catalytic domains and the N-terminus regulatory domain and the C-terminus oligomerization domain, and they're all shown in different colors in this diagram right here. So the catalytic domain contains the active site, which is where the substrate and the cofactors bind. And also contained in the active site is a little orange circle right there, and that is going to be the iron atom, which helps out the reaction. And the oligomerization domain just connects the two subunits.
Moving on to the existing forms of PAH, it typically exists in the resting form or the T state and exists in equilibrium with the activated form. And these are all primarily found in the liver. Here is a cartoon that models how the enzyme is allosterically regulated through phenylalanine. In the resting form, the N-terminus residues typically cover the catalytic site or the active site to which phenylalanine binds to metabolize the reaction. However, in higher concentrations or with consumption of phenylalanine, it can then bind to a regulatory domain, which is located here with the pentagon and orange shaped figures. And that can cause a conformational change in the protein itself, which causes the N-terminus residues to move away from the catalytic site, to which then expose the active site to binding of L-phenylalanine. This conformational change allows for homotropic regulation because L-phenylalanine is both an allosteric activator of the enzyme as well as a substrate to the reaction overall.
So now we're going to talk about the two amino acids that are really important to this enzyme. So there are two histidine residues that allow for interaction in the binding site. The first one is histidine 290, and that one interacts directly with BH4, the cofactor, because it orients its positive charge towards the hydrogens on BH4, which is a positively charged region. And the other residue is histidine 285, which interacts directly with L-phenylalanine because of its aromatic structure, which is oriented towards there as well. These two histidine residues also are important because they bind directly to the iron, which is contained in the active site. And iron is really important to the function of this enzyme because it has multiple oxidation states. And the other amino acid is tryptophan. So there's one tryptophan residue, which is really important to the structure of the enzyme, tryptophan 326, and it's oriented such that phenylalanine can bind to the enzyme very close to it. And because it's also positively charged and aromatic, it provides a stable environment for both the BH4 cofactor and L-phenylalanine. So now we're going to talk a little bit about the structure of BH4 and how it plays a role in this reaction. So as we know, some enzymes need a cofactor in order to function completely. So BH4, which is short for tetrahydrobiopterin, supplies electrons from its double bonds to help PAH convert phenylalanine to tyrosine. And it works together with oxygen to become 4A-OH BH4, and it gets hydroxylated by oxygen. We're going to talk a lot more about the mechanism of how that happens. So this is important because it shows an oxidation reduction, so it can be used again.
So given that BH4 is a cofactor and is required for the metabolism of the reaction, we can delve deeper into a more complex mechanism of how the reaction takes place. And so here is L-phenylalanine and BH4, the cofactor. With the incorporation of an oxygen molecule, it can then hydroxylate both the cofactor as well as the L-phenylalanine to create the product L-tyrosine as well as this 4A-OH-BH4. And later on in the reaction, um, the oxygen that was added on to the cofactor can then be reduced and lost through a dehydration reaction in water to help continue the reaction, but that will be talked about a little bit later on. Right now, we're going to look at a close-up of the mechanism of the reaction in the actual active site of the enzyme. And so here diagrammed, we have a close-up of the catalytic site, which can be found by looking for the orange circle or the iron that is bound to both of the histidine residues that associate with both the iron, the cofactor, and the substrate. So here we can see L-phenylalanine is bound in the active site and is closely associated with tryptophan 326, as well as the histidine 285 residue. And this iron, which is bound to both these histidines and a glutamate, is important for the mechanism of the reaction because it helps to mediate the incorporation of the oxygen molecule into both the cofactor and the substrate. So taking this structure into consideration, we can then move into a even more complex mechanism of the enzyme. And so here we have the essential components for the reaction. Here's the phenylalanine, the cofactor BH4, and the iron with the associated residues on the enzyme itself. So with the incorporation of an oxygen molecule, it can then form a bond through the mediation of the iron and then form a bond with the cofactor BH4, which ultimately will be hydroxylated as this bond is cleaved. And so this intermediate, the 4A-OH-BH4 is formed. However, the L-phenylalanine still is present in the reaction. And so the second part of this oxygen molecule then is attacked by the phenylalanine and is incorporated and becomes hydroxylated further on into the reaction to then become L-tyrosine. And leaving this intermediate here for a OH-BH4, it is then allowed to dissociate from the PAH enzyme. And then in order to keep the reaction going, the regeneration of this cofactor BH4 is necessary. And so to continue running this reaction, the BH4 must then, after it's dissociated from the PAH, is both dehydrated, which will form this water molecule, as well as reduced through other enzymes to regenerate this BH4. And so the BH4 can then bind back to the PAH and the mechanism can continue to run. And so that kind of completes the entire mechanism for PAH. As it starts at L-phenylalanine, it requires the use of this BH4 to help with the hydroxylation of the L-tyrosine as the final product.
All right. So thank you guys so much for listening. We hope you enjoyed this presentation. Let us know if you have any questions.
Thank you.